Photoreceptors in the eye only register isolated hits of light across the back of the retina. Yet our conscious experience is a continuous reality of solid shapes and recognizable objects. Resolving these fragmented signals into coherent scenes requires a specific sequence of physical computations that occur within milliseconds of light hitting the eye. This raster plot maps the activity of a single neuron. As we move deeper into the cortex, we find cells that ignore raw light points entirely, spiking only when the brain detects a specific, abstract identity. The visual system achieves this level of abstraction through hierarchical computation, taking simple inputs and combining them into increasingly complex outputs. We will trace the physical path of this data, starting from the optic nerve, moving into the thalamus, and up through the cortex, to see how the brain converts local light signals into high-level concepts. The visual system operates as a specialized processor, physically translating raw photons into the structured meaning of our conceptual world. After light is transduced into electrical impulses by the retina, the signal exits the back of the eye and travels down the optic tract toward a junction called the optic chiasm. This schematic traces those visual pathways. At the optic chiasm, the data is physically split. 60% of the axons cross over to the contralateral side of the brain, while 40% remain ipsilateral, staying on the same side. The sorted data streams hit their first major relay station, the lateral geniculate nucleus, or LGN. This structure is organized into 6 distinct layers. Layers 1 and 2 make up the magnocellular pathway. These layers house large neurons dedicated to processing high-contrast information and tracking broad movement. Layers 3 through 6 form the parvocellular pathway. The neurons here are smaller, specializing in fine detail and color data. Despite this rigid sorting, the neurons in all 6 layers process data using the same circular center-surround architecture that was initially established in the retina. The brain segregates these data streams by origin and function to prepare for complex geometric scenes. The segregated signals travel from the LGN into the 4th layer of the primary visual cortex, known as V1. This is where hierarchical integration begins. In the 1950s and 60s, neuroscientists David Hubel and Torsten Wiesel discovered exactly how V1 neurons process this incoming data. They observed a shift in the system's architecture. Multiple LGN neurons with isolated circular receptive fields project axons onto a single pyramidal neuron in V1. If those incoming circular fields converge in a precise linear row, they create a new property, orientation selectivity. The V1 pyramidal neuron reaches its action potential threshold only when a line of light strikes all aligned circular centers simultaneously. Tilting that line of light causes it to miss the centers and strike the inhibitory surrounds, resulting in 0 spikes. This chart graphs an orientation tuning curve. The spike rate remains flat across most positions, but hits a sharp peak the moment the bar reaches the neuron's preferred angle. By mapping isolated circular points into a single line, the brain achieves edge detection. It has physically added circular points together to compute geometry. Once V1 detects a geometric edge, it faces a new problem, determining which direction that edge is moving. To solve this, the cortex uses a delay line mechanism. It exploits the varying physical lengths of axons to manipulate the timing of electrical signals. Consider a circuit built to detect left-to-right motion. 3 presynaptic neurons are arranged horizontally, projecting to one postsynaptic cell. Their axon lengths are staggered, the longest on the left, the shortest on the right. As a vertical edge starts moving left to right, it triggers the furthest neuron first. Because of the long axon, that electrical signal takes the most time to travel. By the time the edge hits the middle and right neurons, the delayed signal from the first axon catches up. All 3 impulses hit the postsynaptic cell at the exact same moment, causing a synchronized firing. If the edge moves the opposite way, the signals arrive at erratic intervals. Without synchronization, the cell does not fire. V1 organizes these specialized neurons into dense vertical structures called hypercolumns, mapped by their orientation and directional preferences. Motion is not passively seen. It is calculated using the physical constraints of time and distance within the cortex. After V1 computes basic edges and local motion, the visual data physically bifurcates into 2 specialized processing streams. The first is the dorsal stream, known as the where pathway. It projects upward toward the parietal lobe. Said by the magnocellular pathway, the dorsal stream terminates in area MT, specializing in global motion vectors and visual motor control. Functional MRI data confirms this division. When human subjects track moving dots on a screen, metabolic activity inside area MT spikes. The moment the dots stop moving, the activity vanishes. The second route is the ventral stream, or the wet pathway. It projects downward into the inferior temporal cortex. The ventral stream focuses on form recognition, object representation, and linking visual geometry to memory. Ultimately, the signal physically splits because interacting with an object requires fundamentally different mathematics than recognizing that object's identity. Following the ventral stream deep into the temporal lobe reveals the highest level of visual processing in the human brain. Researchers observed this processing during neurosurgery studies by recording single-unit neuronal activity in patients viewing various images. They tracked the data using raster plots, scanning for dense spikes of activity immediately following an image presentation. In one specific patient, a neuron inside the temporal lobe triggered a sustained spike rate when shown a photograph of the actress Halle Berry. This neuron fired robustly to various viewing angles, different lighting conditions, and even a pencil sketch of her face. The firing pattern held even when researchers flashed the text string Halle Berry on the screen. The neuron fired just as densely, despite the written words sharing no visual characteristics with her face. This reaction was specific. When presented with images of other politicians or celebrities like Bill Clinton, the neuron remained silent. The firing data suggests this neuron responds to an abstract identity, operating independently from the raw photon hits at the beginning of the chain. The visual system deconstructs light into edges and motion vectors, routes them through specialized processing streams, and ultimately reconstructs those fragments into high-level conceptual reality.